Very good evening. 2 minutes, we'll start, okay? Yeah. So, how are you, student? How your studies is going on? How are you reading? Okay. So, let us start the class. So, can you tell me whether my audible and screen is visible, student? Let us start the class. Can you tell me whether audible and screen is visible? Jainvi Chitradurga. Okay, very good. So, let us start the class. Okay. So before going to start the class, here is the question for you. Very, very easy question. Okay. So can you tell me, student, pick up the incorrect statement. Okay. So I think it is a little tricky. Okay. So let us see whether you will answer or not. Okay. So pick the incorrect statement. Sucrose is disaccharide. Option B, uracil is pyrimidine. Option C, glycine is sulfur containing amino acid. And option D, cellulose is polysaccharide. Can you answer me, student, in the chat box also? In the chat box only, you can answer me. Can we answer me? Which is the correct answer here? Think and answer me. Which is the correct answer? I'm waiting for your answer. Answer me, then we can continue the class fast. Can you answer me which is the correct answer, or you don't know means you can tell me don't know. No problem. Yes. Student, the correct answer is incorrect statement they are given, right? No, not a correct answer. Think once again. Not option D. How brilliant you are. No option C, now you'll give the option C, right? Yes, very good, student. Yes. So glycine is the very simplest amino acid, right? Yes. Glycine is simplest amino acid, right? So you can write the structure means it will be like NH3 plus C. Here is the H, here is the R. So R group is also the H only, right? Wait 1 minute. Yes. So R group is made up of the H only, right? So here it is the COO minus, right? So glycine is a very simplest amino acid. When it comes to the sulfur containing amino acid, so it is, can you recall cysteine and methionine, right? Yes. So these are the sulfur containing amino acid. When it comes to the sucrose, very good. It is the 1 minute, sorry. If I tick here, you will get confused, right? Okay. Yes. So sucrose is a disaccharide. Sucrose is made up of glucose and fructose, right? It is the non-reducing sugar. That also you have to remember. Okay. So non-reducing. So when it comes to the uracil, yes, it is a pyrimidine. It is going to present only in RNA, right? Okay. So I have told you about the cysteine and methionine. When cellulose, yes, cellulose is the polysaccharide. It is homopolysaccharide. Homopolysaccharide. A student? Okay. So then coming to the next. So that's why the correct answer is option C, right? Okay. So let us come to the lipids. So what is the lipid, students? Lipids. So lipids, we have talked that lipids are made up of fatty acid and glycerol, right? 1 minute. So we'll call it as alcohol, right? So it is made up of fatty acid and alcohol, right? I'm making again mistake. Sorry. Alcohol, right? Which is the alcohol here, student? Glycerol, right, student? Glycerol. So what is the definition of the lipids? So lipids is the esters of fatty acids and alcohol, right, student? Esters of fatty acid and alcohol, we will call it as the lipid. So you know what is the structure of the glycerol? Right. CH2OH, here only I have given, right? Sphere. CHOH and CH2OH. It is the structure of the glycerol. Student, so if I add the one fatty acid here, okay, I'm adding the fatty acid here for the glycerol. Okay, this is a glycerol, right? So glycerol structure I have written. Now I am adding the one fatty acid. See here, CH2O, C double bond O, R. Okay, because the fatty acid consisting of R, COO, H group. Okay, so that's why it is written as R, C double bond O. Okay, student, see here now, CHOH, CH2OH. Can I call it as, which name can I call it as? Can you tell me what is the name can I call it as? What is the name of this compound? Can anyone try to answer this? See, student. So here, single carbon I have esterified. Okay, this is what we will call it as the esterification. Esterification. Okay, so this is what I will call it as the monoglycerides. Monoglycerides. Okay, for example, if I do for the 2 carbon, CH2, so okay, CH2O, C double bond O, R group, R1 I will write. Okay, see here, CH, O, C double bond O, R2. Okay. CH2, O, C double bond O. 1 minute, sorry. I will write only to this thing. Okay. Triglyceride, they are only given. So that's why I'm not writing this. CH2OH. So what can I call it as? This is the carbon 1, this is the carbon 2, this is the carbon 3, right? So now can I call it as a diglyceride? Diglycerides. Glycerides. Okay, student? 2 here. When 2 fatty acids are esterified here, we will call it as a diglyceride. When single fatty acid, I will call it as monoglyceraldehyde. And see here, they have esterified 3 fatty acids. Here, this is the 1, this is a 2, this is a 3. So that's why they are calling as triglyceraldehyde. Okay, so you have to remember that. Okay, student. So let us come to the structure of the, classification of the fatty acid. See here, classification. So when it comes to the classification, it is based on double bond. Based on double bond. Can you try to answer, student? So how many types are there? One is saturated and another one is unsaturated. Unsaturated. So whenever we are talking about the saturated fatty acid, they do not have double bond. Do not have double bond, right? Double bond, they don't have double bond. Adatra double bond iralla. So they don't have any double bond. Okay. So then coming to the unsaturated fatty acid, student, they do have double bonds. Okay. So they have double bonds. Okay. Okay. So usually saturated fatty acids are animal fat. Animal fat. For example, if you have taken the curd, or you have taken milk, that is animals only, right? You have taken the milk, you have converted into the curd, then you have taken the ghee, right? That ghee is also the saturated fatty acid, right? Dalda. Dalda is also from the animal only, right? So that is also the saturated fatty acid. When it comes to the unsaturated fatty acid, so these are from the plant origin, plant seeds. From the seeds, we will get the oil, right? So these are the unsaturated fatty acid. This is very good for the health. Okay. So why we should not take the saturated fatty acid means, so these saturated fatty acids are solid at room temperature. Okay. So that's why we should not eat this. This is solid at room temperature. Okay. Not eating. I am telling that it is not good for the health. Okay. For example, we have taken this saturated fatty acid in the blood vessel, the fat is going. Okay. So what happened? This saturated fatty acid, we know that what is our body temperature, students? 37 degree. That is room temperature, right? In the room temperature, what is the position of this? It is going to become solid. So it is going to become a solid. See, here is a blood vessel. So this fat is going to be accumulate here. See here, this is going to be accumulate here. Then passage of the blood cells is going to be restricted. See here, how can the blood cell can move here? Because blood cell is carrying the oxygen, right? See here, it is restricted, right? So that is the reason. So saturated fatty acids are not good for the health. Okay. So it will cause the cardiac diseases. It will cause cardiac diseases. Cardiac diseases. Okay, heart attack. So whenever the blood is not going correctly, it is going to cause the heart attack, right? So second thing, we will go to the, in the animal fat only, the another example will come is coconut oil. Sorry, not in the animal fat, in the saturated fatty acid, it will come the coconut oil. Coconut oil. Coconut oil is the saturated fatty acid. Whenever you keep like the coconut oil like that only, it will become the hard, right? It will become a solid. So that is also one of the example. Coming to the all the plants oil except this coconut oil will come under the unsaturated fatty acids. Okay. So let us talk about unsaturated fatty acids. Student, there are two types of unsaturated. One is we call it as the MUFA. MUFA, and another one we call it as the PUFA. PUFA. What do you mean by MUFA and PUFA means? Mono unsaturated fatty acid and polyunsaturated fatty acid. Poly unsaturated fatty acid. So example I will give you arachidonic acid. Arachidonic acid. Arachidonic acid. It is good for health. Okay. So and you can take linolenic acid. Linolenic acid. Okay. So these are comes under the PUFA, polyunsaturated fatty acid. These are essential fatty acid also. Our body cannot synthesize these PUFA because, so in our body there is no enzyme which will put the double bond after the 9th carbon atom. So that's why, so this fatty acid we will take in the diet. Okay. That is arachidonic acid and the linolenic acid. These are the essential fatty acid also. Okay, student, these are essential fatty acid also. Essential fatty acid. Okay. So then other than this, there are the another type we will study about this. Okay. So that is lipid is classified into the two types. Can you recall, student, which are those types you have learned in the, right? You have learned in the 11th. Can you recall? So one is simple lipid, and another one is the conjugated lipid, and third one is the derived lipids, right, student? Okay, let us talk about the simple lipid. Simple lipid means it does not consisting of anything, only the fatty acid chain. Okay, so only the fatty acid chain, that we will call it as a simple lipid. Okay, the example is lipids. Sorry, fats and wax. Okay, fats and wax are example of the simple lipids. Fats and wax. Okay, wax. So coming to the conjugated lipid, so here they are telling that something is conjugated to that, right? Something is there with that, right? So yes, The things which is with that is the glucose. It may be a glucose or it may be a protein. Okay. So for example, if it is combined with the glucose, we will call it as glycolipid. Glycolipid. When it combined with the protein, we will call it as what we will call it as lipoprotein. 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 Okay. This is the thing we will call it as glucose and the protein. So let us talk about the lipid, derived lipid. So this derived lipid is consisting of, this is a ring structure. Okay, it is consisting of the carbon and hydrogen ring structure and the open chain structure. Carbon and hydrogen ring structure and also the open chain structure. We'll call it as a derived lipids. Okay, so the example for the derived lipid is cholesterol. Cholesterol. Okay, so students, our sex hormone, for example, testosterone, progesterone, estrogen. So these all the things is come from the cholesterol. Okay, estrogen, and you can take the testosterone, and progesterone. Okay, so these all the sex hormones are come from the cholesterol. Okay, so this is about the classification of the lipids. So do you have any confusions here? So Jainvi, Chitradurga, do you have any confusions in the lipids? Can you tell me, or whether you understanding this? Okay, fine. You can tell me, no problem. So this is a structure, and here they are given another structure, students. So see here, this is a phospholipid structure. Where you have seen that phospholipid structure? Have you seen in the cell? This is a cell. This is a cell wall. This is a cell membrane. Sorry, animal cell membrane. This cell membrane is made up of the phospholipid, right? Phospholipid. So this is the plasma membrane. Plasma membrane. Plasma membrane is made up of phospholipid. You have seen that, right? So there's a phospholipid structure. So it is a phosphate group and this is a lecithin. Okay. So this is a lecithin group. So let us go to the nucleic acid. So can you tell me what is the nucleic acid, students? All of them know that, right? It is a genetic material, right? It is act as genetic material, right? Okay, let us talk about the genetic material. Whenever we are talking about the genetic material, two things will come. One is DNA and another one is the RNA. What is the DNA full form? Deoxyribonucleic acid. Nucleic acid. When it comes to the RNA, it is ribonucleic acid. Right, student? Okay. So let us talk about this. Okay. So whenever we are talking about the nucleotide, we'll call it as polynucleotide. Polynucleotide. Right. What do you mean by polynucleotide? It is made up of single nucleotide chains. Right. So see here, it is made up of nucleotides. Right. Nucleotide. So if you take the protein, so how the single protein is made up of the amino acids. So like this, so the nucleic acid, either DNA or RNA is made up of the nucleotides, right? Nucleotides. See here, what is the composition of the nucleotides, students? Can you try? Can you try? Nitrogen base plus ribose sugar plus phosphate group, right? Phosphate group. So when it comes to the nucleoside, it is made up of only nitrogen base plus ribose, right? Here you have to remember nucleoside. Side means nitrogen base plus ribose. Nucleotide. Tide means what? Nitrogen base, ribose, along with that phosphate group is there. Okay. So this is about the nucleotides and the nucleoside. Okay. So next, coming to the nitrogen bases. 1 minute. Yeah. So coming to the nitrogen bases. So how many nitrogen bases are there? Nitrogen bases, nitrogen bases. So there are two types, right? One we will call it as a purines and another we will call it as the pyrimidines. Pyrimidines, right? So when you're talking about the purines, adenine, and guanine, and when we're talking about the pyrimidine, thymine. 1 minute. Yeah. So when we're talking about the pyrimidines, thymine, uracil, what is another one, student? Cytosine. Very good. Can you tell me the composition of DNA now? DNA is made up of which is the purine and which is the pyrimidine here? It is made up of adenine, guanine, thymine, and cytosine, right? When we are talking about RNA, it is made up of adenine, guanine, uracil, and cytosine. So whenever they give one nucleotide chain and they will tell you identify whether it is RNA or DNA means you have to check whether it is having the uracil means it is RNA, if it is having the thymine means it is a DNA. Okay, fine. So let us talk about here the structure. So here again is the NH2 group. It is the adenine. You have to remember the structure. Okay, they may give you the structure and they may tell you which is the name of this compound. So that's why. So guanine, it is having the ketone group. See here NH2, cytosine, and in the thymine at the fifth stage, it is having the fifth position. It is having the methyl group. Okay, so the methyl containing nucleotide is methyl containing nitrogen base or methyl containing pyrimidine is thymine. You have to remember. Okay, fine. This thing you have to remember, student. So there is a adenine and guanine. So between the adenine and thymine, there is a double bond, right? Adenine and thymine, there is a double bond. And between the cytosine and guanine, can you tell me between the cytosine and guanine? There will be the triple bond. Okay. So, and which are the types of the bonds you can see in the DNA? So, see here, the bonds you can see in the DNA. Bonds in DNA. So, these are the bonds. Okay. One is the phosphodiester bond. Phosphodiester bond. Where you can see phosphodiester bond? Between the nucleotide. For example, so here is the adenine. 1 minute. 1 minute. Okay. So here, see here, this is the adenine. So this is the guanine. So this is the thymine. So this is the cytosine. Okay. So adenine bond with the thymine and guanine with the cytosine. 1 minute. Okay. So guanine with the cytosine and thymine with the double bond adenine. And this is triple bond with the cytosine. See here, student. So this bond is there now, this bond. So this bond we will call it as the phosphodiester bond. Diester bond, right? Between the nucleotides. And so in between these two, right, the bonding. So this bonding we will call it as the hydrogen bond. Hydrogen bond, right? So whenever the DNA denature or DNA nature, so both the strands will be separated, right? Because of this breakdown in this hydrogen bond. Okay, so this is the hydrogen bond, which is the second bond we have learned. So that is the hydrogen bond, right? So see here, it is the hydrogen bond, hydrogen bond. So can you tell me which is the another bond? N-glycosidic bond. Where is the N-glycosidic bond is present between? N-glycosidic bond. Where it is present, student? Can anyone tell me? Between nitrogen base and between the sugar, right? So glycosidic bond is between nitrogen base and the sugar, right? So the another bond is there in the DNA. So that is the phosphoanhydride bond. Phosphoanhydride bond. Where is this phosphoanhydride bond? You can see. Can anyone tell me? So these are the important bonds, okay, which is present in the DNA. For example, if you see the structure of this thing, 1 minute, you can see here structure. So just I am writing here, okay, this is the nitrogen base. Here you can see the phosphate group, right? CH2, here is O, here is the phosphate P double bond O. So here is the O, P double bond O. So P double bond O. O, right? So this is O minus, right? So here, this is a triphosphate. This structure I am retaining as the ATP. Okay, ATP, if you take the ATP means the structure. So there is a triphosphate, right? Three phosphate are there. One phosphate, two phosphate, three phosphate, right? So bond between these two, okay? The bond between this phosphate group, we will call it as the phosphoanhydride bond. Okay, student? This bond, see here, this sugar and between the nitrogen base, this thing we will call it as glycosidic bond, right? So this is the thing I have told you now, okay? So here between this bond we will call it as a phosphodiester bond. Sorry, this between the here to the another nucleotide, okay? Fine. So do you understood these things? Nucleic acid? Okay, let us go to the structure, okay? So whenever we are talking about the structure of the DNA, it was given by Watson and Crick, right, student? Watson and Crick in the year 1953. Everyone knows that, right? So our DNA is double helix, right? It is the double helix, right? So it is double helix and right-handed double helix. And whenever we are talking about the one chain length like this and another chain like this, right? 5 prime, 3 prime, this is a 3 prime and 5 prime. So what is this? We will call it as student anti-parallel, right? The DNA is anti-parallel in nature. Let us talk about the structure, okay? See here, this is the right-handed helix, anti-parallel in nature. This is the 5 prime to 3 prime. This is 5 prime to, this is 3 prime. Okay. So see here now. So this is 1 turn. Okay. See here. So student, this is the 1 turn. Okay. From here to here, we will call it the 1 turn. It is the 36 angstrom. Okay. 1 turn. In the 1 turn, there will be 10 base pairs. 10 base pairs. Here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Right. So there will be a 10 base pair in 1 turn of the DNA. Okay. So see here, this thing is very minor, right? This groove is minor. So that's why we'll call it the minor groove. And this is larger, right? Larger. This is, we call it as the major groove. So what is the diameter now? What is the diameter of the DNA? So DNA diameter is 20 angstrom. Okay. So student, here you can see the distance between the two nucleotides, right? Here is the one nucleotide and this is another nucleotide, right? The distance between these two is the 3.4 angstrom. Okay, distance between two nucleotides. Okay, fine. This is about the structure of the DNA. Here you have to remember our DNA is right-handed, double helix, antiparallel, and 36 angstrom is the 1 turn, 10 base pair, and major groove and the minor groove, and the distance between two nucleotides is 3.4 angstrom. Okay, fine. So let us talk about the very, very, very important topic, that is the enzyme. So student, what is the enzyme? What we will call it as the enzyme? Enzyme we will call it as the biocatalyst. Right? Enzyme we will call it as biocatalyst, right? So can anyone tell me what is the biocatalyst? Why can't we use in the chemistry experiment? Why we are using only in the living organism experiment? These enzymes, why it not work in other? Can anyone tell me? Can anyone tell me, student? Jainvi, Chitradurga. What do you know about the enzyme, students? Student, this enzyme is made up of proteins, right? These are proteinaceous, right? These are made up of protein. So, but except only ribosome, right? Only except ribosome. Ribosome we have studied in the translation, right? It will use it in the translation where from the using the information present in the mRNA, utilize it for the protein synthesis. It is present in the bigger subunit of the ribosome, right? So this is the ribosome enzyme. It is the RNA, not the protein. Okay. So when we're talking about the enzymes, there will be a simple enzymes and conjugated enzymes. Okay, so simple enzyme, name only telling that it is very simple, right? It is made up of only protein, right? It is made up of only protein. Okay, so when we're talking about the conjugated, something is attached to that, right? Something is going to be help for that enzyme for performing the function, right? Yes, so that is we call it as a conjugated, right? So here, along with the protein, it requires coenzyme, right? It requires coenzyme, or along with the protein, it requires cofactor, student, or protein, or it requires prosthetic group. Prosthetic group. For example, if you take the protein, protein is you, and coenzyme is your best friend, right? Your best friend is sitting beside you, right? So like that. So if your best friend is not there, means you are not going to work. Not like that. I'm giving just example. Okay. So it is like more than a close friends. Okay. So this protein without this coenzyme, it is not at all going to work. Okay. Yes. So that is in the conjugated enzymes. Okay. So you can take a coenzyme, cofactor, and the prosthetic group. Okay. So let us come to the enzyme. So what is the function of this enzyme, student? What it is going to do? So see here, this enzyme having the three functions mainly. Okay. See here, it will increase rate of chemical reaction. It increase rate of chemical reaction. So student, nothing. The enzyme is like catalyst. Okay. So in the chemical reaction, in the chemistry lab, you will add the lead or something as for the catalyst, right? Lead is act as a catalyst, right? It will increase the rate of the reaction, right? So the same thing our enzyme is also going to do. That is in the living organism. Okay. So that is, it increases the rate of chemical reaction. And what is another thing why we are using this enzyme? It decrease the come from. So student, this enzyme catalyzation, whenever this enzyme binds substrate, it is going to release some binding energy. Okay, it is going to release some binding energy. Binding energy. Whenever the substrate and enzyme see here, carbon dioxide and H2O Okay, so see here, student. Without using the enzyme, this carbon dioxide is going to contain carbon dioxide and water converting into the bicarbonate. So it will give 200 substrate. Okay, 200 with the 1 hour. So carbon dioxide is there, hydrogen is there with mixing, which is going to give the by 200 product. Okay. Okay, some network issues there, sorry. Okay. See here, in presence of the carbonic anhydrase, carbonic an anhydrase, carbonic anhydrate, they say, so that is, you can see here, millions per second. Okay, 6 millions per second. It is going to convert this substrate into the product. Okay, so that is the main thing. So it is going to, what here happen? It is going to increase the rate, and see here, the same carbon dioxide and water, the product is also same. It is not at all going to change any substrate into the product. Only it is going to change the rate of the reactions, right? So that does not change. Yeah. So another thing you have to remember here, the first enzyme, okay? The enzyme name was given by okay, sorry, student. Okay, sorry. One minute. So, one minute. Here I have told you this is the active students, some network issue is there. No problem, we'll continue the class. Okay, I think now it will not happen. Okay, fine. So, 1 minute. Yeah, so I have told you, right? So the substrate is going to be converted into the product. Now the enzyme is free now. Then again it can bind to the substrate and it can convert this into the product, right? So see here, I have told you that this is the absent of the enzyme. So when the enzyme was absent, it is going to take so much of the activation energy, right? The activation energy is very high in absence of the enzyme. So when our enzyme is there, the activation is energy very low. See here, the activation energy is very low. So I have told you because this is because of the binding energy, right? Because of the binding energy, I have told you, right? Yes. So this is about, you have to remember, the activation energy is very low in presence of the enzyme. Fine. So I have told you that the enzyme is going to bind to the substrate. The enzyme substrate complex is going to be formed, and the enzyme and product complex is going to be formed. Then the enzyme is going to separate and product is going to be separate. So now what will happen? This enzyme again bind to the another substrate and it will form the enzyme substrate and enzyme product, and it is going to release the enzyme plus product, right? So this is how the enzyme is going to be work. Okay. Enzyme substrate, here you have to remember, student. Enzyme separate, after that enzyme product. Okay. Then enzyme is separating and product is separating. Okay. Fine. So here you can see the lock and key model. So lock and key model was given by the Emil Fischer. So according to this model, they have told this active site. Okay. The active site of the enzyme was rigid. Okay. This active site of the enzyme was rigid. Okay. For example, if you take the, see here, this is the active site they are given, right? So this is a very rigid. It is for only for the particular substrate. For example, if you take the lock, okay, for particular lock, you have to use a particular key, right? You can't use some other key. If it is the case, means everyone have the different key, means everyone can open the every home door doors, right? Yes, doors lock, right? So this is how the particular substrate only can bind to the particular enzyme, and that will convert into the product. The other substrate cannot match with this active site, right? So that's why they are given that this is very rigid. Okay, the active site was rigid. Okay, particularly the substrate, for example, if you take, this is the structure. This is the structure of the enzyme. Okay, this is the structure of the enzyme. This is the active site. Active site. Okay, so the substrate should be like this only. Okay, the substrate should be like this only. Then only it can come and bind here. Then it can convert this into the product. Right? So this is what they have told in the lock and key model. Okay, so for the particular lock, the lock here is the enzyme. Okay, so particular lock enzyme, there will be a particular substrate. Okay. Okay. So whenever the substrate come and bind to the enzyme, so it will, the enzyme substrate complex is going to be formed, then it is going to form the product, right? So here you can see the structure. The structure is similar here. Then it can, the active site is specifically made for this substrate only. Then it is going to bind to this, right? It is going to bind to this and going to convert this product, right? So this is what we call it as lock and key complex. For a particular lock, there will be a particular key, right? Yes. Okay. So then let us talk about the substrate concentration. See here, in the substrate concentration, student, is a substrate concentration. Here the substrate concentration is goes on increasing. For example, 2, 4, 6, 8, and 10. Okay, so here you can see the velocity of the reaction. Velocity means which speed the reaction is going to be taking place, right? See here, whenever we are increasing the substrate, there is an increase in the enzyme activity, right? Enzyme activity. See here, sorry, enzyme velocity. 1 minute. Enzyme velocity. Enzyme velocity, right? So increasing the substrate concentration, increasing in the enzyme activities or enzyme velocity, you can also call as. See here, when it was increasing at the particular stage, when enzyme concentration is equal to the substrate concentration, it reaches the maximum. Okay, it reaches the maximum because I've told you that whenever the enzyme was free, when it converts the substrate into the product, it will convert to the another substrate and it will convert that into the product. So student, so whenever the chemical reaction we will do right by using the enzyme, we will take the enzyme concentration very less. Okay, we'll take the enzyme concentration very less and we'll take the high substrate concentration. Okay, so for example, if you take the enzyme is very less here, we simply adding the more substrate means the enzyme is very busy in converting the other substrate, right? So what happened now? So here the enzyme concentration is going to be exceeded. The 1 minute. So here what happened? See here, the enzyme become less. Okay, the enzyme is less to convert substrate. To convert substrate means what happened here? All the enzyme is occupied by substrate. Occupied by substrate. If you add more substrate also, there will be a reaction velocity is same here. So there will be no change will be going to happen because the enzyme is very less here. Simply goes on adding the substrate means how can this enzyme is going to convert into the product? It will also take time, no? So that's why, so it takes the maximum velocity. It reaches the maximum velocity here. So this is what we will call it as Vmax. Vmax means nothing but maximum velocity. Okay, student, maximum velocity. See here. So how can you take the Km? What is the Km? Km means Michaelis-Menten equation. Michaelis-Menten constant. Michaelis-Menten constant. Okay. So Km. So how can you calculate the Km? Nothing. So which is the number you have got for the Vmax divided by 2. Okay. Km is Vmax divided by 2. Okay. If you do that Vmax divided by 2, you will get the Km. Okay. Student, remember here, the Km will be the whenever Km is high. Okay. So whenever the Km is high, the enzyme is having the low affinity for the substrate. Low affinity for substrate. Okay. So whenever the enzyme Km was very less, okay, it is having high affinity for the substance. For example, you see now. So now you can tell me the enzyme A, okay, it is having the 0.001 and enzyme B is having the 0.1. Can you tell me which is having the high affinity and which is having the low affinity? B is having high affinity, right? Because it is having the low Km value. Sorry, 1 minute. So B is having low velocity. So low affinity. It is having low affinity, right? So A is having the high affinity. High affinity, right, student? See here, A and B enzyme I have taken. So which is 0.01, which is having the high affinity because the Km value is less, right? B is having the 0.1, that is the high Km than the A. So that's why it is having the low affinity. Okay. So high affinity is very much required for the enzyme to convert product. Okay. So let us go to the effect of the temperature. So student, our enzyme is act in the very narrow range temperature. Okay. If the little temperature was high also, if the little temperature was low also, it is not going to work. Okay. If the pH was high also, low also, it is not going to work. Okay. It requires optimum temperature and the optimum pH for the working. Okay. So here, after that, it will decline. Okay. It will decline above and below the optimum temperature. Okay. So what happened at the low temperature? If the low temperature, the enzyme will be inactive. Okay. For example, if you take the enzyme is active at the 37 degrees Celsius. If I keep that in the 20 degrees Celsius, is that going to work? No, it is inactive, right? Yes. So see here, at the high temperature, the enzyme is get denatured. We know that our enzyme is made up of the protein, right? If I give it in the high temperature, means what it is going to be happen? The tertiary structure is going to be damaged, right? So when it is going to lose the tertiary structure, then it is going to be denatured. All the bonds is going to be broken down. For the enzyme activity, it must require the tertiary structure, right? Okay. So see here they are given the graph here. So this is the optimum temperature. So this is what the optimum temperature where the enzyme is active here. And this is the optimum pH. See here, below that is also decline, and above the pH is also decline, right? So here it is going to be work at the optimum pH. Here I have taken the example of pepsin. Pepsin is going to work in 2 pH, in the acidic pH, it is in the stomach, right? When you take the amylase, it is going to work in the 6.7 pH, it is going to be in the saliva, right? Amylase. So when you take lipase, what is the pH of lipase? 12. It is going to break down into the lipids. It break down the lipids, right? So in the exam, this may be asked you like this, student. See here. So they are given the graph here. So they will not give the enzyme name here. Okay. So they will give the 2, 6.7 and 12 like this. Okay. So you have to remember at which pH this enzyme is going to work. Okay. So see here the classification of the enzyme will go. So remember the classification of the enzyme oath lil. One is the oxidoreductase. This enzyme is involved in the oxidation and reduction, and transferase enzyme. So this is involved in the transferring. So they see here is the one reactant. From here to here, it is transferring right here. So here is there in the S group. So it is going to transfer into the S dash, right? This is transferring, right? So that's why we'll call it as transferase enzyme. The hydrolase enzyme, which will break the products here. AB is there. It will break the AB into, okay? It will break the AB by utilizing the water. The enzyme which will break down the any products by using any substrate into the products by utilizing the water. We'll call it as the hydrolase enzyme. It will break down the substrate into the product by utilization of the water. See here, it is breaking the AB into the AB with the utilization of H2O into the AOH and the BH. Okay, student? Okay. So let us come to the lyases. Lyases will also do the same thing only, but without the water, it is not utilize anything, but it will break down here. XCCY. It will break down into the XY and C double bond C by utilization without utilizing any water. Okay. Lyases will not use water. Hydrolases will use the water. Then coming to the isomerases. Isomerases, student, what is the main function of the isomerases is? It will change the position of the substrate. Okay, it will change the position of the substrate. For example, you see here A, B, C. In the first position A, in the second position B, in the third position C, right? So what it is done now, see here. Now B in the first position, C is in the second position, and A is in the third position, right? It will group exchange reaction. Okay, group exchange reaction will done by the isomerases. Group exchange reaction is done by the isomerases reaction. So then coming to the ligases, you know, right? In the biotechnology, we have learned about the ligases. What is the function of the ligases? It will join 2 nucleic acids, right? So the function is ligating molecular suture, right? Or glue, we will call it as, right? Yes. So it will add C here, X plus Y by utilizing the ATP. Student, here you have to remember the ligases require ATP. Okay. So with the help of ATP, it is joining the X and Y, 2 reactants into it is going to be join the 2 reactants XY. Here ATP is utilized. So that's why you are going to give the ADP plus PI. Okay. So remember oath lil, oxidoreductase, transferases, hydrolases, lyases, and isomerases and ligases. Ligases is at the last. Ligases require ATP. Okay, students, for joining the 2 reactant into the product. Fine. Okay. So do you have any confusions here, student? Let us go to the inhibition of